Hello, everyone, and welcome to GemPharmatech's exclusive webinar on the Knockout All project, or KOAP. My name is Koming, and I'll be your moderator for today's session. We're thrilled to have you join us as we explore one of the most ambitious, large-scale mouse genetic initiatives in biomedical research. I want to briefly introduce our company, GemPharmatech. GemPharmatech is a global leader in genetically engineered mouse models, with over 25,000 strains currently in stock, supported by a team of more than 1,500 scientific experts. Our infrastructure spans 7 subsidiaries across China and the US, with large-scale SPF vivarium capacity and over 6,000 new models generation capability. We are also able to support more than 1,000 preclinical studies per year, covering immuno-oncology and beyond. Whether you need breeding, custom model design, or full in vivo studies, we provide integrated, scalable support. Our newly expanded capabilities in the US position us to partner more directly with biopharma teams, navigating early discovery through R&D enabling work. For today, we're excited to have Dr. Bingzhou Han, a key leader at GemPharmatech, walk us through the latest progress, insights, and applications from this groundbreaking project. Now it's my pleasure to introduce today's speaker, Dr. Bingzhou Han, who is our head of our genome editing platform at GemPharmatech. Dr. Han has played a pivotal role in designing and executing the KOAP, overseeing the development of high-throughput gene editing platforms and their applications in generating disease-relevant models. Dr. Han, the floor is yours.
Okay, thank you, Komo, for introduction, and thank you everyone for attending this webinar. So let me start my presentation today about the genome editing platform and their applications in GemPharmatech. So as we all know that animal models are in vivo models that are widely used for research on human disease because we cannot do some experiments in human due to ethical problems. And among all these animal models, mice will be one of the most important ones because it is a mammal and of great importance. We know that mice consumes about over 70% of the experimental animals consumed over the years of biomedical research, and it contributes to 67% of the Nobel Prize in Physiology or Medicine. And from science, it contributes to 22 out of 50 of the major breakthroughs in biomedicine over the last century. So why is mouse so important and feasible as a model vertebrate or model mammal? Because in all the mammals, it shares a relatively close genetic relationship with human because it's a mammal model, although not as close as non-human primates, but also in all the mammals, it has the most developed and applied breeding skill and also experiences and also the most advanced application of genetic tools, including transgenesis, genome editing, and other approaches. And also, through the clear breeding for over a century, there are clear genetic background annotations for all the inbred and outbred strains of mice all over the world. It makes us easy to do comparisons among different strains and also some MHC-based studies. And also, last but not least, the small body size, relatively short lifespan, and strong reproduction capacity make us easy to grow into large population or colonies of mice and do comparison experiments, set of different elegant designs to do mechanism studies and drug evaluations. So from the basics of model animal mice, so we need to obtain mutants for gene function study. So from the development of genetics, we know that there are 2 basic ways for us to obtain mutants. The first is forward genetics. It originates from spontaneous mutations during breeding, or random induced mutation through radiation or chemical ways like yin-yu, or biological ways like gene trap or virus trap. So in these ways, when we get the random mutations and then we have the phenotype of our interest, then we can study the phenotype and also to do mapping to identify the causing gene and then to set up the causing effect of the gene and the final phenotype or the disease. In this way, it's so-called forward genetics from the phenotype to the genotype. And years later, after the development of targeted modification strategies, including embryonic stem-based targeted mutagenesis and endonuclease-mediated genome editing, then people can target the genes in the mouse genome and then to get the mutants of the certain genotype and to see the phenotype of these homozygous mutations. And this approach from the genotype to the terminal phenotype is so-called reverse genetics, because it is developed later and in the reverse direction of forward genetics. However, due to the understanding of the central dogma from the DNA to RNA to protein and to the final phenotype, the approach of reverse genetics itself is exactly the way how genes interpret themselves and to contribute to the final phenotype. So the development of genome editing technology actually takes more than half a century and many milestones, including starting from the discovery of DNA double helix by Watson, Crick, Franklin, and their colleagues, and then also the development of Sanger sequencing by Sanger DNA sequencing by Sanger, and with the understanding of some of the important genes and also their sequence, and then finally, in 1982, so scientists developed this embryonic stem cell-based homologous recombination in the cells, and then several years later, they developed into heritable mice. And this is the first generation of targeted genome editing, which makes it possible for us to target some genes and to disrupt their functions, using embryonic stem cells. However, since the restriction of ES cells to limited species and limited genetic backgrounds in mice like 129 and Black 6N, so years later, after the development and application of the zinc finger nucleus, then the application of the genome editing technology has been expanded to multiple organisms, and also broke some of the restrictions of genetic backgrounds. And then years later, after the application of TALEN nucleus and of course the powerful CRISPR-Cas system, we can now confidently see that we have broken most of the barriers for mouse genome editing, and we can do elegant and versatile editing and strategy designs to for different approaches. So we can have a basic have a simple introduction about the CRISPR-Cas system, since I believe that most of our audience is very familiar with this. So it is composed of the nucleus itself, Cas9, and also a single guide RNA, which is responsible for recognizing the target site by Watson-Crick base pairing between the genome DNA and also the sgRNA in the alternative version. And also there are two domains in the nucleus. One is the RUVC domain, the other is the HNH domain, which is responsible each for cleavage of one of the double strands of the DNA. And then the cleavage by the Cas9 nucleus guided by the sgRNA results in a double strand break with a blunt end in the genome. And then in mouse, the double strand break can be repaired by endogenous pathways, either the end joining pathway or the homologous directed repair pathway, which is usually activated in dividing cells in cell cycles. So let's first look at the end joining pathway. So with the end joining pathway, it is usually error prone and may introduce mutations in random insertions or deletions in this site. So if there's no donor DNA in this system, and then there will be uncontrolled insertions or deletions in this site. So if we design this target and make the double strand break in some coding regions, the uncontrolled indels may introduce frame shift mutations in this coding gene and cause a simple knockout effect. In fact, this is the easiest way for the nucleus-mediated gene knockout. However, this way has some certain drawbacks, like the potentiality of introduction of truncated proteins and cause of knockdown, or even maintaining the gene's function. It is less used these days. And also we can introduce double-strand DNA donors through the end joining pathway and insert this into the genome. However, this integration will be in any orientation, so it is not easy to predict. And also due to the error-prone characteristic of end joining pathway, the junction site, the sequence of the junction site will not be precise. Thus, it is not the most elegant way for us to integrate exogenous DNA. The homologous directed repair, on the other hand, generates precise insertions of exogenous DNA because it is homologous directed, and the homologous arm will be precisely repaired, causing the insertion or substitution of the sequence inside the two homologous arms in the donors, either single-strand donor or double-strand donors. And these ways we can achieve DNA replacement including like point mutations or insertion of small-sized DNAs, including logs-Ps or sequence coding protein tags, or other, like insertion of large DNA sequence, including exogenous coding sequence, reporter genes, query recombinase, and other regulatory sequence, including promoters or transcription termination elements. So we can see that after the development of endonucleases, especially CRISPR-Cas, we can dramatically shorten the timeline for getting heterozygous mouse from like pronucleus injection of the endonuclease system, and we can shorten this time period for at most for 3 months, because we can reduce the process for embryonic stem cell culture and screening process, and also because we don't rely on the embryonic stem cells, we can break the barrier for mouse genetic backgrounds, which is difficult for the embryonic stem cell generation, like Black6J or immunodeficient backgrounds, since like non-SCID or NSG, NCG, etc. Instead, we can do the microinjection based on these backgrounds and generate mutants. And also, the efficiency of CRISPR-Cas-based editing is very high, of course. But however, so the embryonic stem cell-based targeting still has its own application, especially for the multi-step editing and also insertion of very large fragments targeted, like hundreds of thousands of base pairs. So based on this background knowledge, so I want to have a brief introduction about the mouse genome modification strategy we usually use these days. So the modifications we can do includes like knockout includes the conventional way or conditional way, and also knocking of different exogenous genes, including coding sequences, regulatory sequences, point mutations, reporter genes, and tag coding sequences, etc. Also, we can combine these modifications together to generate complex modifications, like targeting multiple genes simultaneously and do gene humanized modification or substitution. Last but not least, although not considered as a traditional like genome editing technology, the transgenic technology for overexpression of genes is also powerful and is applied in those certain circumstances. So as I mentioned before, so the conventional knockout is the basis for most of the genome editing. As I mentioned before, the strategy for like exon frame shift mutation is less applied because it's easily to generate like truncated mutations for either N-terminus or C-terminus truncated proteins through different ways, either like infirmity G or like functional like shortened transcripts. So instead, now we tend to use multiple CRISPR-Cas target sites in the introns flanking one or multiple of the essential coding exons, or even using target sites outside of the gene body, like upstream of the 5' UTR and downstream of 3' UTR, to delete partially the essential exons or even the whole gene cassette, to generate a knockout. So in this way, we can generate deletions up to millions of base pairs and can delete tandem genes, sometimes some gene clusters, like the hemoglobin gene clusters or the serpina A1 to A5 gene cluster, which is responsible for the R1 trypsin inhibitor protein. So conventional knockout itself is very powerful and helps us to study some of the gene's functions. However, sometimes some essential genes knockout may lead to embryonic, prenatal, neonatal death of the mice, which is not wanted for some of the studies for like adult, like aging or metabolism or something else. And also because some expression of some genes are ubiquitous or are strong in different tissues and organs. So when the knockout phenotype is complicated, like multiple tissues or organs are affected, it is difficult for us to like tell the primary effect or the secondary effect in this case, or we cannot 100% rule out the crosstalk between the cells or tissues. And also, last but not least, if there's functional difference in different development stages or in the adulthood or aging in these mice, and we do a conventional knockout, we can now tell the contribution of these genes, expression in different stages. Thus, a more powerful way is needed for more dissected gene function analysis. That's why we need to, like introduce the conditional knockout. So conditional knockout majorly is, based on the application of a Cre lock system, which is originated from bacteriophages. So the Cre recombinase can mediate the recombination of two lock species in the same orientation, and then cause the recombination of these two lock species and the removal of the sequence within these two lock species in the like linear DNA, like the chromosome. So in this case, we need to construct a so-called flux allele with two lock species flanking one or multiple of the essential coding exons of the genes of interest, and then this allele can be generated by CRISPR-Cas-based knocking using one or single donor or two independent donors by homologous directed repair. And then this flux allele is successively generated, and after mediated deletion, this gene will be converted into a null allele and then achieve a Cre-dependent knockout. So let us take this as an example. When we breed these mice into a target gene homozygous of the flux allele, and also together with a tissue-specific Cre driver made by knocking or genesis, we will mention later, and then get this Cre positive flux homozygous mice, and this Cre recombinase can mediate the recombination of LOXP and knockout of the gene of interest, like the gene Y, in the certain tissue with Cre expression. Like in this case I mentioned here, so we use a adipose tissue specific Cre driven by the adiponectin Q promoter. And we can see that this target gene Rep18, the protein is gone in different adipose tissues, including the BAT and VAT, but not deleted in the liver. So which means that this breeding can result in a adipose tissue specific knockout of Rep18. Additionally, by fusing a estrogen receptor ligand binding domain with mutation to the Cre recombinase, so this protein can be a like time-dimensional inducible protein, because the estrogen receptor ligand binding domain, so it naturally binds to the heat shock protein 19 in the cytosol. So this fusion protein is kept into the cytosol and kept away from the nucleus and the genome, that's genome DNA, in normal circumstances. However, when we give a like estrogen like ligand, which is tamoxifen or 4-hydroxytamoxifen, the binding of this ligand to this mutated estrogen receptor ligand binding domain will release this protein from heat shock protein 90, and this protein will be translocated then into the nucleus and contact to the genome DNA and mediate LOXP recombination. So which means that this recombination in this recombinase Cre-ER is a ligand tamoxifen dependent inducer of recombination. So which means that we can administrate a time-dimensional control of this conditional mutation. Then we have already seen that how we can make strains like flux alleles with flux alleles, and then let's see how we can make other alleles like Cre recombinase expression or other like expression of other exogenous genes like reporters or proteins. So we can do this by knocking exogenous operating frame or coding sequence to the endogenous gene. We can either do the knocking in the 5' and the 3' of this gene. And also when we do the knocking into the 5' we can include a transcription termination signal like the polyadenation sequence to inhibit the expression of the endogenous gene in this way, and then to disrupt the function of the endogenous gene. And also in other cases, we can maintain the function of the endogenous gene by using different linker proteins or two peptides to separate the translate, or then using two peptides to separate their translation products, or use arrays for separate translation of the upstream protein and the downstream protein in this case. And also the strategy is based on our demand of the expression level of the exogenous gene and also the final consequence we want. And also we can also substitute some of the coding sequence by a point mutation or like small precise deletion or insertions to make certain disease models in this case. So one of the applications of these knock-ins can be humanized genes. We can see that using one or multiple target sites, we can substitute the whole mouse gene with a homologous human gene with certain exons and introns, and to make a full humanization protein. However, this strategy is very elegant, but since most of the genes are very long, or say on average like 30 kilobase pairs, so the knocking will be less efficient. So alternatively, we can choose to use a mini gene strategy to knock in the human homologous genes coding sequence or the cDNA of this human gene with the transcription termination signal to the 5' terminus of the mouse homologous gene. And in this case, this human gene is driven by the endogenous promoter and is expressed as the pattern of the endogenous mouse gene. And in this case, we can also do a full humanization in this case. And also, this insert is much smaller compared to the intron containing full human gene and is more efficient and easier to construct. Last but not least, for some of the especially membrane proteins like immune checkpoints, we don't need to fully humanize the gene. Instead, we only need to humanize the extracellular sequence of these genes, like for evaluation of the antibodies against these targets, for binding of this extracellular domain, and then for the transmembrane and intracellular domain, we need it to be maintained in the mouse form to mediate a normal downstream signal transduction. In this way, the exons coding the extracellular sequence of mouse can be substituted by the homologous human genes extracellular domains coding sequence in this case, which result in the expression of a chimeric human mouse protein with an extracellular domain of human and also the intracellular domain and transmembrane domain of the mice. In this case, it can also simultaneously achieve the binding of the antibody drugs against this target and also a normal downstream signal transduction. So this strategy is feasible for multiple immunocheckpoints, including PD-1, TIGIT, CTLA-4, etc. So other than knocking, transgene is another strong way for us to overexpress exogenous gene in this case. So we can have this construct with a promoter, a gene of interest, and a reporter, which may indicate the expression of this exogenous gene. And this construct can be released for macroinjection in the embryos and then for random insertion. So this strategy is also feasible for bacterial artificial chromosome insertions for hundreds of kilos of base pairs. So for precise regulated expression of the exogenous gene. And also if we modify this plasmid and also back, we can do simultaneous overexpression of multiple genes or very fine-tuned expression of fluorescence proteins or query recombinances. Also, this traditional way can be further enhanced by utilization of transposons like piggyback or sleeping beauty to increase the efficiency of transgenesis. But if we use transposons to mediate transgenesis, the copy number of this transgene is usually less compared with the traditional way when we use linear DNAs to do random insertions. However, in other ways, so the insertion of this transgene, the copy number and the insertion site throughout the chromosome will be random. So then after we get the founder mice in this timeline, so we need to separately breed these founder mice to background mice and to carefully evaluate the inheritance of the transgene among generations for establishment of a stable germline. So in order to avoid the time-consuming stable line construction process, we can alternatively choose to insert the promoter and exogenous sequence, the so-called transgene cassette, into safe harbors in the mouse genome, including the ROSA26 in the 6th chromosome and the H11 site in the 11th chromosome, and other safe harbors like Tegri. So in this case, we can avoid the time-consuming stable line construction precise, and also because this is this is a targeted knock-in, so the genotype will be clear and it is easy to do genotyping and validation of this insertion. And also the individual difference among generations and among the siblings will be limited. However, because the target site integration site of this so-called super TG is certain, we cannot obtain strains with different expression levels. And in case when we need to very strong expression of the exogenous gene, we still need to go to transgenesis way to do this generation in sometimes. So last, what I want to emphasize about the point mutation is that most of the human genetic variants associated with disease are single point mutations, and due to the high similarity between mouse and human coding gene, so we can easily correspond some of the protein coding point mutations to the mouse gene, and then a single point mutation will do to mimic the human SNV and to see the disease consequence of this mutation and also generate the model for the mechanism study. So, but practically, usually we need to introduce an irrelevant silence mutation to avoid the replicative cleavage of Cas9 when we generate these models to increase the efficiency. So, based on these capacities and also our technicians, in GemPharmatech, we are capable to generate more than 6,000 models new germlines every year. So from your innovation or your requirements, we can help you to to design the strategy and to polish it. After negotiation and verification, we can do the generation for you normally within 6 months and to deliver your customized genome editing mice with an in-time rate for more than 69%. Well, sorry, more than 96%. So based on our capacity to generate genome editing mice, so in 2018, we have started an ambitious project called the Knockout project. So we can tell from the name that our aim is to generate knockout for all the protein coding genes and flux allele for these protein coding genes in the mouse genome, which is near 23,000 of genes. So now we are halfway of this project, so we have completed the generative knockout of 11,000 of them and also more than 10,000 of flux. And also for the application in combination, we have independently developed more than 240 Cre strains for different tissues and organs. So the strains are all made on Black 6J background, which is the most commonly applied genetic background for genetic or genome mutation research. And it is easily for you to to do combination with your background mice and also all the other query lines you have for your resource. So, sorry, during this process, we have become one of the largest vendors repositories of the world for the mouse germline collections. So for easy approach and convenient search about these models, other than the gene name or strain name or strain ID, we have separated these strains according to different gene families or signal pathways for your easy to search. So this is a simple about the AMPK pathway or insulin related pathways. So every of this single ball will link to the knockout project's strains of this target gene or protein. So during this process, we have optimized and improved our technology in different aspects, including automatic conditional knockout strategy and sequence design to accelerate our process for generating the new strains. And also, we have optimized our LOXP template length and also the distance between these two LOXPs to increase the generation and also the LOXP recombination efficiency of these strains. And also, we have combined the electroporation and macroinjection together to increase our capacity to generate more models. And also, in our macroinjection, we can simultaneously generate knockout and conditional knockout of the same gene, and we have set up a workflow for simultaneous screening of these two germlines and maintenance of them. So also, for tissue-specific knockout and lineage tracing, especially for the application in combination with our flux alleles, you have developed more than 240 Cre strains, and over 40% of them are validated by ourselves by different approaches or by publications. When we validate our Cre strain, we will breed them with flux reporter to achieve a lineage tracing consequence and to like faithfully recapture the actual Cre consequence when we use this in combination with other flux alleles. And this terminal activation is indicated by the expression of the fluorescence downstream of this flux cassette. And this is evaluated by different approaches, including fluorescence photomicrographs and immunofluorescence in combination with different indicators of different cell types, and flow cytometry analysis, which is majorly used for immune system related queries like T cells, B cells, or myeloid cells. And also, this is an example for validation of one of the strains, the PDX1 query, which is known as a query specifically for pancreatic cells. So you can see that we use this flux reporter as a tool for validation, and the expression of TD tomato red fluorescence indicated the query activity. As a result, we can see that although most of cells of the pancreas express the red fluorescence, some of the cells in the enterostomach or the duodenum also displayed an expression of the TD tomato fluorescence. So which is not strange, because the PDX1 expression is earlier than the terminal differentiation of the pancreas. So it is expressed in some of the progenitor cells in the endoderm, which may differentiate into pancreatic cells, or in the enterostomach or duodenum cells, some of them. So the fluorescence will truly, faithfully recapture the actual activity of this query during the whole development and growth process. So here's a simple summary of the delivery period of our strains. So we have kept more than hundreds of our KOAP mice and the query tool mice as live mice status. So for these mice, we can manage the delivery to your facility within 1 month. And also, however, for the other majority of the strains, we have preserved these mice in like sperm or embryo status, like from F1 or later generations. So for these mice, if we can manage the the IVF or other process, and then resuscitation of the embryos, and then deliver the individuals within 4 months. And also, if it is from founders, the sperm from founder male, we can also manage the delivery within this same timeline. So for delivery of sperms, usually we need additional 1 to 2 months to first have the F1s or the additional live mice, and then to collect sperms from these individuals. However, for the strains, if we have sperms or embryos in stock but not from founders, we can also manage the delivery of these sperms or embryos within 1 month. So for those strains we have not developed yet, so for knockouts we can manage the generation within 6 months. But for the other kinds like flux or knock-ins, we can manage delivery within 8 months. So as I mentioned, so the in-time rate of our newly generated models is overall more than 96%. I believe it's like most cases we can manage the delivery, so as we guaranteed. So our knockout project library have supported more than 600 publications by the end of last year, with a total impact factor over 7,800. So we have supported like publications on different like important journals like Nature and Science and Cells, and some of their like major subjournals. And also we have supported the publication in different scientific facility or universities, not only in mainland China, but also in North America and Europe. Other than these ready-to-use mouse resources and the knockout and flux for gene function study, we have also originated different disease models from the knockout project for different fields, including metabolism, autoimmune diseases, and genetic diseases. So first, for obesity, we know that these two doctors, the Professor Coleman and Fredman, they have independently separated the OB and DP mice, which are later characterized as the key players in the leptin pathway this mutation will lead to the obesity of these mice. However, other than this, we have like this LMS1 knockout in our KOAP library, which encodes a centrosome protein which involves in celiac regulation. The mutation of this gene in human is highly related to Alström syndrome. So we have carefully evaluated the phenotype of these mice, and we can see that these mice develops a spontaneous obesity, which is not less compared to the OV mice, and also it displayed a higher blood glucose level compared with the OB mice, and also a higher insulin level as measured. Interestingly, we have measured the food intake of these mice in different time points. We can see that the average food intake of these mice is higher compared with their black 6 wild-type littermates, which means that this model is more mimics the like human obesity, because normally in human the fat people or the people with obesity tend to intake more food compared with like the population. Also, this model, other than the spontaneous obesity and hyperglycemia, this model also displays a spontaneous hypercholesterolemia and also non-alcoholic fatty liver disease. We can see that in these knockout mice, the level of total cholesterol and HDL and LDL are all significantly increased at 20 weeks, and also the liver function is impaired with like strong staining of oil ride and also fibrosis stained by serous ride, which means that 21-week-old MS1 knockout homozygous mice spontaneously develop into NAFLD. Also, other than the glucose or lipid-related metabolism, the knockout of UX itself is a good model for hyperuricemia, because this UX is expressed in most mammals to prevent hyperuricemia, to convert the only soluble urate into easily soluble allotonin. But this gene became a pseudogene in human and some of non-human primates. So if we knock out this gene in a mammal like mouse, it loses the capacity to convert the urate into allotonin and leads to hyperuricemia in this case. We can see that in this knockout model, the protein of UX is gone, and also the level of the serum uric acid is significantly increased in either male mice or female mice. And also this increase of blood serum uric acid level can be partially reversed by the treatment of allopurinol, which is an anti-hyperuricemia drug in human, which can inhibit the conversion of theanine to urate. Also, KOAP models can also contribute to generating the disease model for systematic lupus erythematosus, because we know that one of the major causes of SLE is the activation of autoimmune pathways against double-stranded DNAs. So also these models, if they have an increased like B cell level or activation of B cells, it is very suitable for the evaluation of treatments against B cell. So you can see that the loss of DNA in exonuclease TREX1 will lead to the increased innate immune against nuclear DNA, and finally an immune hyperactivation of SIGA-STING pathways, because the double-strand DNA in the cytosol or the extracellular environment will be increased due to the loss of this exonuclease gene. So it may lead to a like SLE-like phenotype in the mice. So in the practical analysis of this model, the TREX1 knockout display no protein expression of this protein, of this exonuclease, and also there's an increased anti-double-strand DNA antibody level since 7 weeks old mice, and also there are significantly increased ALT, AST, and LDH level, which means that the liver and heart function of these mice is impaired. So there are systematic, also other like pathological abnormality of these mice systematically, which indicates a SLE-like phenotype, and it's suitable for evaluation of different B-cell targeting drugs, and we have successfully supported different cases through this. Finally, so we know that rare diseases, like a major health threat for some of the populations, and is raising more and more concerns about the whole world. And also due to the development of CGTs, we now have approaches to like treat some of the rare diseases and to complete like the cure of them to benefit these minority people. So the majority of the rare diseases are genetic diseases, but they are highly diversified for their causing mutations. But the majority of these causing mutations are loss of function mutations or hypomorphic mutations. So which means that if we know the causing gene of this genetic rare disease, we can use knockout or conditional knockout models to mimic the disease. So so we have developed like more than 60 of the disease models in GemPharmatech, and we can see that most of these models are originated from knockouts or conditional knockouts, like the models for this autosomal dominant polycystic kidney disease, or like for some arginine deficiency or other mutations related to the urea cycle, and also some of the models of the PFIC. So this is a part of the summary of the disease models, genetic disease models we originated from knockout projects, including phenylketonuria, Fabry disease, Niemann-Pick disease, or thalassemia, or others. So we have now validated more than 50 models from the KOAP as genetic disease models, and now we are seeking to do more in this process, especially for eye diseases. So based on our resource, so we have collaborated with one of the best hospitals in China, which is the Beijing Union Hospital. They have a rare disease treatment. They set up a rare disease treatment and diagnostics innovation development institute in the last year, and 10 different facilities or companies have joined together to set up this institute. So we are very glad that we can set up this united laboratory of rare disease animal model and drug efficacy evaluation with them, and contribute to the research and also model generation and also drug screening and evaluation for rare disease patients. So as one of the example, so one of our thalassemia model like NCGX has become a gold standard for non-clinical evaluation of beta thalassemia cell or gene therapies against beta thalassemia. So we can see that we have supported more than 6 of the like IND applications in China for CGT against beta thalassemia. So some of the clients choose to buy our mice and do the experiment themselves. The others tend to like do the experiment on our side and do in collaboration, like a collaboration with the CRO company. And also our hemophilia A model F8KO has supported evaluation of RNAi therapy against different targets like heparin cofactor 2 or other factors. So this is the like end of our today's presentation. So last, let me introduce our company again. So we offer a one-stop solution for biomedical research, including different genetic modified mice models and also models related to these genes, including like surgery or drug-induced disease models or like germ-free mice. And also we can provide services based on these disease models for different pipelines, including oncology, autoimmunology, metabolic disease, cardiovascular disease, neurobiology diseases, and also genetic diseases. So based on our capacity, we are very glad to be at your service and to solve your biomedical problems or to help your development of your drug or therapies together. And also, as the summer will be in, so we are now having a 50% off of our knockout project models, including the knockouts, flocks, and cray. So please take advantage of this exclusive order through the end of August. And if you are interested, please contact us after this webinar or visit our website or contact our contacts. Thank you all for your attention, and your questions are warmly welcome.
So thank you, Dr. Han, for that very insightful and comprehensive presentation. We will now move to our Q&A session. If you have any questions, please type them in the chat or the Q&A box, and we'll address them as they come in. So I have several questions right now that entered, and maybe we can begin with some of them. So Dr. Han, can you discuss the rationale for including both constitutive knockouts and conditional knockouts in the project?
Yes, thank you. So as I mentioned, so sometimes like conventional knockout is powerful and it can help us to dissect some of the gene's functions. But sometimes so a detailed analysis for the gene function of different tissues is very difficult to like study. And also sometimes there are unrecognized situations where the gene's function will be like analyzed or like occurred. So let me take one thing as an example. So like the genes I've mentioned, like ARG1 or ASS, which are key players for the urea cycle. So it's normally expressed in the liver for metabolism of like nitrogen-related amino acids. However, so we may know that these genes are very important in liver. However, in these days, we have seen researchers, like manipulating these genes in different immune cells because they have seen that in some of like the tumor microenvironment status, some of these related pathways are activated in some tumor-infiltrated leukocytes or myeloid cells, and this activation of these cells tend to inhibit some of their anti-tumor, like capacity. So this is some new discoveries which we are not commonly aware of. So this is the reason why we need these like conditional alleles to be constructed, and then we can study their function, like in different circumstances, like under different disease models or surgeries or stresses.
No,
This is
thank you very
one.
much for that. Yeah, thank you very much for that. So definitely, it seems that it's very dependent on the study design, what you're trying to study,
Yeah.
whether it's a constitutive knockout may or may not, and a conditional knockout may apply in that situation. Well, that's
Yeah.
a fantastic answer. Thank you so much. The second question we have is, what are some of the biggest technical hurdles you have encountered while targeting such a large number of genes, and how have you successfully addressed them?
So the first one is like how to stabilize our platform for the model generation. So we tend to have we need a like a higher like pass rate or efficiency for like if we do the design and injection or electroporation for once, we can have the models ourselves. This is the first challenge, and we can easily we didn't we have tried different ways. We have gone through some difficulties, but we have done this by like practices and like optimizing the dose we use, and also the like some of the injection amount, and then we have optimized all the system. And then the second one is like the control of the cost, because we started this project when we started this company. We need to carefully use every budget we have. So then after we have like stabilized our platform, then we need to like limit our like colony size or injection, the number of embryos when we do this application. So based on the data we have limited the injection like from hundreds of embryos to like 80 or eventually 70 or 60 of the injections once. So in this case, so we can have the success rate increased to over 80% of this injection to get this mice. In this case, the cost is successfully controlled. The third question may be like the priority thing, because we have like tens of thousand genes for target. So which one should we do first, and then we need to set up like a algorithm for the priority of these genes. So we combine these things together with like research focuses, and also the like the detailed annotation of the gene and protein sequences and the sequences and functions, and we have integrated them together with an algorithm, and to have the list of the top 2,000 and top 5,000 genes, and then we choose to like target these genes first to cover the like research hotspots these years, and then expand the generation of these models to a larger, to a more larger population. Sorry.
No, no, thank you very much. So it definitely seems that, to generate all these kind of knockouts, you need to have a pretty well-established infrastructure. And I think, the audience, the attendees are, I'm pretty sure you're aware that how difficult it might be to create a knockout model. And I think it just kind of alludes to the fact that at our company, we are well equipped to help you, generate the knockout model that you want. So thank you very much again for highlighting that, Dr. Han. Can you highlight a few examples of disease areas where the KOAP-derived models have had the biggest impact so far?
So for disease research, I believe that with the highest, I believe that so at least in mainland China now, so the ones of highest impact will be the like rare genetic disease models, because these model resources is limited globally, especially in China. So we have like evaluated the feasibility of these models and the phenotype of these models, and also some of the like therapeutic approaches like diet-based therapeutics, small molecules, or even genetic therapies based on these models to demonstrate the feasibility for these models for if drug efficacy evaluation. And we have collaborated with different scientific facilities or industrial partners to see if they can help to facilitate some of the development of drugs or therapies. And definitely they will finally benefit some of the minority patients.
No, that's great. That's definitely, I think, that's the whole purpose of our KOAP, to help, advance biomedical research on a whole. Definitely very great. So are there collaborative opportunities available for, I mean, I guess you kind of told us about a little bit after your previous answer. Are there collaborative opportunities available for academic or industry partners to co-develop models through KOAP?
Sure. So we accept in the collaborations like majorly for two aspects. The first one is tool mice, like for the fluorescence reporter expressing mice or the query recombinase expressing mice with some like uncommon promoters, but of great importance. If they have no evidence supporting this, we can co-develop these models. We can offer a special discount for these models, and then we generate them together, and we can discuss how how we distribute the intellectual property of these models. So in the like detailed consult. And also the other way is that because commonly our knockout project does not include non-coding genes. However, if there's one client who's interested in some non-coding genes which is reported to be like important in cell models or other based models like for microRNAs or non-coding RNAs, so we are happy to like generate this knockout of this microRNA or non-coding RNAs coding gene if possible, and include them in our knockout project library and with the same price, so which may reduce the like the cost of the researcher and increase our library, increase and like add one new strain to our library.
Yeah, definitely sounds like a very advantageous situation to and all our partners in academia and as well as industry. Another question that came in is, I guess again, kind of alluding to your previous question, the previous questions we got is that do you envision this resource being integrated into a larger functional genomics or drug screening platform?
So frankly speaking, so this is a very good question. So for the for the functional genomic analysis, we already know that so our company has collaborated with the other like great facilities or research organizations for the MPC, so the International Mouse Phenotype Consortium, this project. And then we have contributed phenotyping for some of these knockouts. However, like because phenotyping itself needs a very like standard workflow, and also it's very time and labor consuming. So we tend to need to have like a priority for like studying the gene's function with these all the mutants. So we have the priority of these disease models first, and then hopefully if we are capable of doing more of them, so we may finally contribute more to the functional genomic studies. And also for the drug screening platforms, so other than the disease model we have, so we can combine these knockout models with some of like the antibody humanization platforms in our company or other companies. We can combine them together for easy selection for the and for the easy selection for antibody drugs against human targets, but not mouse targets sometimes, and to avoid like cross reaction in this case. I don't know if this answers your questions.
Yeah, I think it pretty much does. Thank you so much for your response. I think that's all the questions we have so far. So maybe we can again thank you again, Dr. Han, and thank you everyone who has joined us today for this very stimulating discussion. We hope this session has helped spark some new ideas on how you can leverage the knockout and conditional knockout mouse models in your research. If you'd like to learn more about our KOAP or request custom models, please visit our website or reach out to our team directly at sales@genpharmatech.com. We'll be sharing a recording of today's session shortly, so please keep an eye out for this email on your inbox. And I want to thank you again all for attending, and we look forward to supporting your next discovery. And have a great day.
Thank you.
Yep. Bye-bye.